again, I'm very happy to be here in opening another Global IMMUNOTALK. It's unbelievable, but this is already Global IMMUNOTALK number 31. And so I'm grateful to be doing this with the co-organizers of the Global IMMUNOTALKs, Dr. Carla Rodlin. And today we are with our fantastic speaker, Dr. Ido Amit from Weizmann Institute in Israel. So as always, just a reminder that from the very beginning, our goals with this seminar series were to inspire immunologists across the world in an egalitarian manner and to increase opportunities for scientific learning without traveling, making the talks inclusive, accommodating, and environmentally friendly. And we want to thank one more time all the speakers that have contributed to this initiative, and remind you that next week, Sasa Enlobu from South Africa will be speaking at the Global IMMUNOTALKs. And so with that, Carla will now introduce Ido.
Thank you so much, Elena. And it's indeed my real pleasure to introduce today our global immuno speaker, Dr. Ido Amit. So Ido was born in Israel and earned his PhD at the Weizmann Institute in 2007. And after that, he came to the US for his postdoctoral training at the Broad and at MIT. And it was there where he made significant contributions to our understanding of regulatory networks that control transcriptional responses in immune cells. And after only four very successful years, he returned again back to the Weizmann Institute, where he started his independent lab. Nowadays, Ido holds the Eden and Stephen Romick Professorial Chair. Now, we all know Ido because of his truly pioneer work in single-cell genomics and big data analysis. His work has made use of these approaches to reveal the cellular complexity of the immune system and also how this changes in multiple disease settings. I'm sure we're going to learn more about this today. We know his work has had tremendous implications in understanding autoimmune diseases, neurodegenerative diseases such as Alzheimer's, and even cancer. Now, because of his significant contributions, Ido has received numerous awards and recognitions. I'll just name a few here. He is the International Research Scholar of the Howard Hughes Medical Institute. He's also the recipient of the EMBO Gold Medal Award and has been elected as a member of the European Molecular Biology Organization, known as EMBO, in 2017. And Ido is also one of the co-organizers of the phenomenal Next Gen meeting, which is truly highly recommended. I hope they come back with that soon. So I'm sure we are up for a treat with his Global IMMUNOTALK, which is entitled The Power of One, Immunology in the Age of Single-Cell Genomics. Ido, thank you so much for accepting our invitation.
Thank you, Carla and Elena, for making this possible for us to connect, even in this challenging year, and for organizing this. It's a great joy and pleasure. I'd like to start by actually toasting. I think the immunology field and science in general has made a huge contribution to these very challenging times, and we don't take time to actually celebrate enough these achievements. So let's toast for immunology and for better years and for the great achievements of all immunologists out there. So thank you.
And cheers. Fantastic. Thank you. Thanks to Ido.
As we celebrate the tremendous achievements of immunologists, we also want to remember all the fundamental areas of research that have been significantly impacted by the current pandemic. And we also raise our glass for a very quick return in a very strong way of every area of science. Thank you. I have to drink. I brought champagne. Okay, now, before we start with Ido's talk, we always have a special question. And Ido has truly delighted us with tremendous technology advances. So before we start, Ido, we would really appreciate if you could share with us, how did you develop your interest in new methods that have advanced so much our science, our scientific understanding?
So, so the way we come to the technology development is usually from the immunology, from the immunological question. We start with a problem. We have a question that we cannot answer with today's technology, and that always what initiates sort of a curiosity. What if we can develop a technology that solved that problem? So with what if challenges, it's always usually you know, disasters. Usually the technology don't work or take a long time until they reach a version that is applicable to drive immunological questions forward. And obviously what comes out are only the actual technology that work, but we have a lot of sketches and different version of technology that we hope would come out soon and make a real impact on understanding immunology. And I'll try to also discuss some of what might be the future challenges, what might be technology. If we had them, we could have solved major questions in immunology today.
Oh, that's wonderful. Wonderful. Thank you so much for your efforts. I very much look forward to your talk and learning more about the details of those future technologies. So with that, maybe then we can get started. Let's see if you can share your slides.
Okay, how is this?
It's perfect.
Perfect. So again, thank you very much for allowing me to speak in this beautiful global immune talk and getting us together even virtually. I'm going to discuss a little bit how technology can change the way we understand immunology and what might be a future perspective of technology that will drive immunology forward. I want to start with what is the mission that my lab is trying to achieve, and that is to have a detailed blueprint of immunity, that is to understand all of the cellular components across the different tissues, both in health and in various pathologies and infection, understand the signaling pathways, and most importantly, understand the mechanism of action, how these different components and signaling and interaction come together to proper immune response or a pathological one. And if we discuss this, I think it's we must take a very deep look into the past and understand that cellular immunology started long time ago by the identification of cells that engulf debris or various pathogens, and these are the macrophages. And in general, if we start thinking about it, later on we started using molecular definition, the ability to define surface antigens to describe the different immune cell types and properties. But this got very complicated when we started looking across different tissues and different environments. So describing in this very deterministic receptors on cell surface, the different T cells and their activity became rather challenging. And I think we should maybe look back and think about our definition, because maybe some of our definition are inhibiting better understanding of the immune system and how it's working through both physiological and pathological scenarios. So cell types unarguably are the building blocks of life, but in the current form, the description is somewhat flawed. The way we try to overlay various cell surface markers to describe all of the immune subsets, it's challenging because it lacks consistency. And cell types, if we think about it, have been historically defined by function, as we've seen with the Mechnikov description of macrophages, morphology, the molecular composition of the cell surface markers, ontogeny, and so on. But none of these single attributes has served to actually define cell types. The history of the cells matter, the location, as we know, matter, the external environment is important, the relation with other cell types, I'll discuss that deeper later, and the future potential of the cells deeply matter. So what we've tried to do is to move from this vague and unquantitative description to initially snapshot, we have to recognize it's snapshots of genome-wide molecular footprints, and these can start to lay a foundation of reproducible cellular coordinates or cell states, as we call them. And the workhorse or the engine to actually driving this is a technology that we call massively parallel single-cell RNA sequencing, or MARSEC for short. And this is basically coupling the capabilities of the FACS to sort individual cells, and during that measure their protein intensities. And as the FACS are getting better, we can measure more and more such proteins. And together with that, put every individual cell, sort every individual cell into an individual well, and then use a completely automated pipeline to generate from these libraries and analysis to come with a ab initio transcriptional identity map, that is identifying the cells based on their transcriptional identity. And this was successful initially to really or we were initially successful in showing that this type of classification, although we're only looking at RNA and a few proteins, is sufficient to describe the various cell types that we have in the tissue. So this is the first study that sequenced a complete tissue without the need for markers, and really could define as good as what we knew given the markers, and even define the various subsets of cells we had in the spleen much better than the classification we had at that time, really showing that this approach of defining the cells based on their transcriptional footprints is a very valid approach to move forward and understand immunology in much deeper resolution than we had before. Now, the reason that this is so powerful and so effective is the fact that we can actually take this data that we collect from different systems, and I'll describe them, and put them on one quantitative manifold that allows us to ask many, many questions. And this is really the power of this technology, that if you do it properly, you can really remove many of the effects of experimental effects and so on, and put all experiments collected out there or in the lab for a very long amount of time, and really compare different immune subsets, their pathway and so on throughout different conditions and perturbation and so on. So this really allows you to do very effective pathway discovery. So understand within these different subsets, what are the different genes and pathways which are activated on these different subsets of immune cells. Understand how genetic perturbation modify the activities of some of these subsets. So again, waiting or looking for an immunological outcome may be very difficult. It may be in some specific tissue or location and may be missed. But if you look on the genetic level, this is highly sensitive to understand such perturbation using single cell approaches, and without them it was extremely difficult to do this. We can start to understand dynamics. Of course, marker-based approaches are very challenging to look at dynamics. So various dynamical systems, and many of the immune processes are dynamical, can be better understood using single-cell approaches. We can start to understand evolution. So if there is no need for markers, we can look at the same subset of immune cell across many different organisms and learn a lot of immunology through this approach. We can better understand cell-cell interaction. I'll focus a little bit about this approach, really identifying and understanding what interaction are happening and how these interactions are modifying the different subsets of immune cells. Understand and combine immune signaling and metabolism with the various immune phenotypes, understand better host pathogen interactions, better define, and these many of the single-cell studies now are focusing on understanding human pathology, understanding which subsets of immune cells are perturbed throughout the process, and last but not least, understanding how the different immunotherapy approaches that we are using today, how are they really modifying the immune subsets and the different activities of different immune cells. Understanding the mode of action of these therapies can lead to the next generation and improved therapies that will be even more efficient than the ones that are used today. So there are many publications that we've had along the way to really use single-cell to all of these critical challenges to better understand immunity, and I'll try to get through the talk to several of these challenges. But I think one thing that is really driving forward the use of single-cell technologies in immunology is the development of new technologies that allow us to look in better resolution, so improving the quality and yield of the single-cell analysis, improving the analytics, but also enabling more technology that look at different properties of immune cells, and I'll describe a few today. So one very important process that we are missing in this snapshot is the time. And obviously immune processes, all of them are dynamic, and understanding these dynamics is really critical. So there are many efforts to use new technology that allow us to look into time, understanding the spatial organization of the cells, so how these units are actually laying in the, what's the coordinates of the different activities of the immune cells properties, better understanding cell-cell communication, and I'll really focus today talk on this aspect, understanding signaling and metabolism, clonal relationships between different immune subsets, being able to use genetic perturbation and understanding the clonal expansion of the cell and the epigenetic state. So there's many new layers of information that one can add into the classical, now called classical, although the whole field started 10 years ago, transcriptomic data, but I think these are going to be very important to better understand the function of the immune system and answer many classical immunological questions. I'm going to focus today's talk on these two technologies, the better understanding of cell-cell communication and signaling and metabolism. So if we think about cell-cell interaction, many of or major immune decisions are actually formed by such interaction between various subsets of immune cells in the tissue, and along with this, the major or the most effective immunotherapy drugs are actually blocking or enhancing such interaction between subsets of activating or potentially pathological immune cells. So really understanding in situ this communication between various subsets of interaction cell is a major challenge and question in immunology today. And in order to answer this question, Amir Giladi, a PhD student in the lab, who is actually finishing his PhD and going soon to his postdoc, and Meirav Cohen, a postdoc in the lab, who's opening her own independent lab these days, and I recommend you to look up her website, and she will have a fantastic lab, I'm sure. Started to think about how can we understand the role of immune signaling and interaction in lung development. So what are the roles that the immune system is playing in modifying lung development? For this, they made an atlas of all of the immune and stromal cell in the lung throughout lung development. But they wanted to ask beyond generating an atlas is definitely not the goal, but really to ask, how do we understand signaling and interaction between various immune and stromal cells, and how does this shape the immunological features and the structural and functional features of the lung. And what they did next is to ask which ligand and receptor pairs do we identify interacting between the different subsets of immune and stromal cell. And to make a long story short, because this work was already published, what they found is that through the secretion of IL-33 of 82 cells, they modify the activity of basophil who become lung-resident basophil, who in turn secrete many different cytokines which are important for alveolar macrophage development. So this whole approach of using the data and then the cell-cell interaction network allow them to use intuition to identify these different connections. But then what Merav and Amir thought, this is again still using much of the knowledge out there and using a lot of intuition and selection, or taking this huge screen and selecting only a subset of the interaction, and then focusing the rest of the research from this signaling map into actually functionally characterizing this interaction. So what if we could have a deterministic method to really find the in situ interaction within the tissue? And these are usually, can be an extremely rare event that can be missed by any approach, just sample all of the tissue. So what they came up with this idea that one can perhaps capture these rare events by simply sorting cases where you have markers that appear on the same cell that should never be on the same cell. For example, a CD11C marker combined with the TCR beta marker. So markers of myeloid cells and markers of T cells. And the evolution of this technology came as such that in order to apply it, one needs to initially sort the singlets, the individual cells of the DC components or one component, the individual cells of the T cells or the second component, and then the very rare cases when they co-occur on the same cell or actually the same peak. And this potentially may help us understand how or what are the interactions that are happening in the tissue in different conditions and how they modify the activity of the cell. And I'll try to go a little bit deeper into this. So this is just an image of some of the peaks that they sorted. And obviously there is a lot of calibration that has to go into this in order to dissociate the tissue on one hand, but on the other hand, to preserve the close interaction of two individual cells. And the manuscript actually describes how to use different approaches and calibrate different conditions until one actually sees an optimal range of such interaction. And once that happens, one starts to see interaction between these two separate entities. And in addition, we had to make an algorithmic solution to actually analyze such data. And the idea we had is to take the individual cells, and that's why one needs to sort also individual cells from the two representative cell types, and then use the computer to actually simulate all possible doublets, anyone that could happen between the individual cells. And that then allows us to see which of all of the potential doublets is actually occurring. And obviously that allows you to remove many technical artifacts, because if all you see in the data are just the most abundant cell in the tissue that are interacting, that's likely not a real interaction, but something technical, and not a real enrichment over what you expect. One can later on use such data and deconvolution approaches to separate the molecules and separate them to their different entities, either the dendritic cell and T cell in this case, and understand how such interaction can modify the expression. And for this, we developed another solution, and that is to understand, and what we saw is that the addition of the two cells basically has a linear expression, and from this we can deduce the expected total UMIs of the molecule in the cells if we had an artificial doublet, meaning if this is not a true interaction that modifies the activity, and from this deduce what the interaction is actually modifying, how these two singlets, when they interact, how we would see a different set of activity of various cytokine signaling molecules, receptors, and so on and so forth. And so, for example, what I'm showing you here is an interaction between dendritic cell and T cell modifies these two different subsets, and one can see upregulation of many different molecules which have to do with co-stimulatory molecules and activation. And this is in an in vitro T and DC experiment, and this is obviously very amendable for in vivo work. And in this case, when we look at the pathogen infected mice and we take out the ear lymph node, what we see again is many interaction between dendritic cells and T cell, again focusing on this technology, and then the changes that are occurring are reminiscent of what we know about activation of dendritic cell co-stimulatory molecules and checkpoints. So some of these molecules are known, others are actually very surprising and interesting. And I label a few of them here, but there are many more. And this is really a very powerful approach to start to understand such interaction and how these signaling events are modifying the activities of both the T cell and the dendritic cell. So seeing that this technology is so effective to parse and understand in situ interaction, we started collaborating or joined forces with Miriam Merad from Mount Sinai, together with Amos Tanai. And what we wanted to understand is which interaction are formed within human non-small cell lung cancer. And to do this, we looked both in the interaction that are formed within the tumor microenvironment and in the adjunct potentially not involved tissue. And we did the same strategy here. The markers that we use is a bit more complex in order to capture all of the myeloid components without contaminating any of the T cell types. Obviously, the human tumors are a bit more complex. And also use T cell markers, again, being able to only look at the myeloid subsets as individual, the T cell subset, and cells that represent true interacting peaks. And this is a map showing you all of the singlet that one identified both in the, from I think at this point we had 10 patients or so, from both the myeloid subsets and the T cell subsets within the tumor region and in the adjunct tissue. And this map basically shows the correlation that we see between any of these subsets and how they are defined within the, or enriched in the tumor environment versus the normal tissue. So as expected, cytotoxic T cells, inflammatory monocyte, naive T cells are seen much more frequently in the healthy tissue, whereas exhausted or Tregs or exhausted or dysfunctional CD8 cells are seen mostly within the tumor. Now looking at the interaction between myeloid cells and T cell, what we found is one very specific population of cell that is both highly enriched within the tumor fraction, as you can see if we compare them, the normal tissue to the tumor fraction, and also highly enriched in the peaks compared to the singlets. And this is also shown in quantification here. And this cell really intrigued us because it was a CD4 positive T cell which was also PD-1 positive and CXCL13 positive T cells. And you can see again, highly enriched in the TME and in the peaks compared to the singlets. We then went on to actually define this new and very intriguing T cell subset, which we named as THT, or tumor helper T cell. And what we found that this T cell subset contains both many of the checkpoint molecules seen on exhausted or dysfunctional CDH, such as PD-1, TIGIT, CTLA-4, and so on, but also many other markers which are not reminiscent of CD8 as expected of CD4 or on Treg. So some of these markers are labeled here, and many others are not. What we additionally found that these cells, now that we can take a closer look into them using their molecular markers, are actually found in usually within these immune hubs, or what people call tertiary lymphoid structures in the TME. And most often, as we find in the peaks, they are interacting with dendritic cells, and more specifically with DC-LAM specific dendritic cells. These are the MREG dendritic cells that Miriam Mirad has defined within lung tumors. What's further extremely interesting about these tumor helper T cells, that similar to the exhausted or dysfunctional T cell, they're highly clonal within the human tumors, and that they don't share a clone with any other of the, obviously the CD8, but also the Tregs or other cells within the tumor compartment. So they seem to be highly clonal cells that do not share with any of the other subsets. In order to better understand how such CD4 tumor-specific cells are formed, we try to generate a mouse model to better understand these cells. We use the B16 ova syngeneic tumors and injected OT2 T cells, and then later on induced ova vaccination to again see whether such cells are also formed in a tumor antigen specific way within a mouse, and then dissected a different region, either the tumor itself, the tumor draining lymph node, or other lymph nodes. And basically what we found is that this THE signature is highly conserved. I'll show you in the next slide, but also it's very specific to the draining lymph node initially, and then later on it can be found primarily within the tumor, as we find in patients. And again, these tumor helper T cells are extremely conserved, except for a few molecules within both human and mice subject, and again, they're specifically localized to the tumor and draining lymph node. So again, as we see in the human subject, these cells are also highly interactive with the myeloid compartment, and specifically dendritic cells, and upregulate many different molecules, suggesting that they are recruiting and activating various myeloid subsets within myeloid and other immune subsets within the tumor. For example, XCL1 is important attractant for DC1. And as we see in the human, they're also highly interacting with dendritic cell in these tumor models. The next question we wanted to ask is, what is their properties across a large database of different tumor models? And we've looked at this recent publication, which looks at a very broad range of different tumors in single cell resolution. And we've tried to understand how these THD subsets look across the tumors. What we found, as expected in this database, that the dysfunctional CD8 T cell, the exhausted CD8 T cells, are very much correlated with the amount of somatic mutation we find in the different tumors. So one would have the highest level in melanoma, and then lung, colon, and liver, and then much less in the other tumors. And as expected from the fact that exhausted T cells recognize the neoantigen of the tumor. But the THD show a very similar profile. Again, one see them highest in the lung, melanoma, colorectal, liver. But for a reason we still do not understand, they also show an extremely high level of T cells in the pancreas. Again, this is still an open question we do not understand. If I were to switch gears and move to a second technology, and that is to look at immunology beyond the cell surface markers, technology that we developed in order to start to understand how we can associate different immune functions such as metabolism, expression of various transcription factor or signaling, and how this associates to different subsets of immune cells according to their transcriptional profile. In order to do this, again, this is one of the technology that took several years to in order to resolve, because of the very large challenge to, on one hand, fix the cells and permeabilize them, and on the other hand, preserve the quantities of RNA. And again, this was done using a combination of methanol fixative with all kinds of salts that preserve the RNA and deactivate the different protein that degraded the RNA. And once one has this platform of a cell, you can very intuitively introduce various antibodies that label different signaling components, metabolic component, transcription factor, and then run it through either plate-based platform, which we like, or the droplet-based approaches, and then compare the different signaling subsets that appear as compared to the global transcriptional landscape, and understand which type of cells are being modified or activated, and so on and so forth. And this a beautiful study and very fun one was really spearheaded by highly talented PhD students in the lab, Jonathan, Fadi, and Adam, together with Asaf Weinel, who's a staff scientist in the lab, and Ido Yofe, a postdoc in the lab. And initially what they started doing in order to understand the technology is to see whether it can indeed enable us to use a wide range of antibodies labeling intracellular activity. In this case, we looked at P38, phospho-P38 intracellular fluorescent intensity, and this is the insect labeling compared to methanol fixation, PFA fixation. All of them allow you to label the fluorescent intensity of the phospho-P38, and DSP, which in our hands at least, we lose the complete signal. And what's really beautiful about this fixation technology is that it preserves between 100 and 1,000 times more RNA molecules as compared to methanol, PFA, or DSP. It allows you to actually preserve the cells for a long time in even 4 degrees, and then run them on your favorite platform. And in our hands, we seem to lose in a range of 40% of the molecule as compared to fresh cells. We then tested whether these fixed cells are actually changing the different distribution of different cell types. And again, this is a 10x run of bone marrow DC culture. Here are the first bone marrow disease, insect fixed bone marrow disease, and one can see this basically retains the same subsets of cells, and they are defined by the same molecule. So we don't see a large shift in the different molecules, except for the loss of a very small molecule. And this is also true when we apply it for more complex environments like PBMCs or different tissues. So again, same result. If we use fresh PBMC, we basically get the same cell proportions. In order to characterize and see that this technology really works for labeling various transcription factors, we use the FOXP3 RFP mic, either sorted using the the RFP channel, or fix the cells and then use a FOXP3 targeting antibodies, and again sorted these two populations, ran them on a 10x platform, and by and large, since I see I'm a little bit running over time, what we see are the same distribution of cells, slightly better enrichment with the FOXP3, but again, 70% or so enrichment using the insect technology, and by and large, we capture the same properties of Treg cells. This is also true when we use the same technology in PBMC. So here we use the FOXP3 antibody from PBMC blood. And again, you can see how all of the cells look like, either in fresh insect, or when we use the FOXP3 targeting antibody, how beautifully we enrich for the Treg population from a very minor fraction to a few percent to almost 60% of the population. And one can take it into a whole very large combinatorial transcription factor network. And when we do this, look across many different many different transcription factors defining different T cell subsets. TCF7 as the more naive subsets, ID2 as the more cytotoxic activated memory subsets, and FOXP3, one can start to see different combination of T cells that surface markers can definitely not delineate, and identify, again, different subsets of T cells that I think are going to be very interesting to understand different activities and pathways related to these subsets that currently have no markers. It also allows to go into immune subsets that lack a clear marker. For example, we looked at FOXP3 Tregs from different sites in the body, from tumors and versus lymph node, and start to understand which molecules and markers best represent these different populations. This can also assist in generating targets for better perturbation or targeting in immunotherapy regimes, Tregs and other immune subsets. Last but not least, one of the areas that we had a lot of difficulties is to define the different myeloid subsets and activity within tumors, and especially define immune suppressive activities within the tumors, these lineage vague immune regulatory cells, or MDSCs, which were characterized by a very vague or let's say very wide markers defining very diverse different myeloid subsets. And if one carefully looks at the literature, the best classification of these myeloid suppressive cells is actually coming not from any of the extracellular definition of these cells, because these are extremely wide and define almost any myeloid subset, but actually the metabolic activity, which have to do with various metabolic enzymes such as arginase 1 and O, and so on and so forth. So in order to better classify them, what we attempted to use here is to take the entire tumor, use the insect technology to fix and permeabilize the cell, and then use these different metabolic enzymes to try to define different activities and markers that define these activities within the myeloid subsets. And what I'm showing you here is the use of arginase 1 antibody to define arginase 1 positive cells. Again, these are cells that define these suppressive myeloid cells in this tumor, and then use different approaches to sequence these cells. To make a long story short, we define different subsets which express very high levels of arginase 1. These are basically defining two major myeloid population. One is the tumor-associated macrophage, and two of these less well-defined cells, myeloid suppressive cells that we call MREG1 and MREG2. Again, they're defined by these high level of expression of arginase 1, especially MREG1 and MREG2 population over here. MREG2 population over here. And again, many other markers that define these different population, like PDPN, low for CCR2, very high for GPNMB, and TREM2. And TREM2 especially intrigued us because we've been working in the lab for quite some time to understand the role of this signaling, this very unique signaling pathway, which is starting to appear as a very major immune signaling hub, which is induced upon pathology. It's hardly found in the normal tissues, both in human and mouse. We see very few subsets that express it, microglia, a few subsets in the hair follicle, bones, and so on. However, upon pathological cases such as neurodegeneration, obesity, fatty liver, atherosclerosis, and tumor, one sees major reorganization of this pathway and expression of TREM2 within many myeloid and subsets, including monocytes and macrophages and so on. And what this study suggested that it might be also involved in regulating or suppressing the activity of the immune compartment within tumors. To make, again, a long story short, since I see I'm starting to run out of time, what we showed is that these TREM2 positive MREG cells are highly suppressive, so the most suppressive myeloid cells that we found within the tumor, much more than any of the classical classification of MDSCs, and they are dependent on TREM2 activity. So if we use TREM2 knockout mice, these MREGs very significantly drop in their quantities. And as a consequence, we also see a very dramatic changes in the immune compartment, very significant immune dysfunction with the TREM2, which is reactivated without the TREM2. So without TREM2, we see infiltration of more inflammatory monocytes. We see a reduction in the dysfunctional CD8 cells and upregulation of various T cell subsets or cytotoxic T cells and NK subsets within the tumor. As a consequence of this, what we see that indeed within the TREM2 knockout, there is a very robust, within the TREM2 expressing myeloid cell, a very strong immune escape mechanism, which is removed by the TREM2 knockout. In a back-to-back parallel study by our long lasting collaborator Marco Colonna, he has shown very nicely that combination of PD-1 with depletion of TREM2 genetically or within antibodies or with use of TREM2 blocking antibodies very significantly impacts the survival of the tumor in many different, both in MCA model, MC38, and other models. So just to conclude and round up everything, what I showed you is PEAK-seq, which is a new and what we think is a very exciting technology that allows us to start looking at in situ cell-cell interaction and characterizing them. What I showed you is a molecular characterization of conserved T helper cells associated to tumors, what we called THT, and they express many modules which of genes and checkpoint associated with dysfunctional CD8 cell. They strongly interact with DCs and are highly clonal within human tumors. High level of these THCs associated with tumors with large number of somatic mutation. And in the second part of the talk, or the last part of the talk, what I described, INSEQ, a new technology for recording single-cell RNA-seq and intercellular protein activity. Again, there's many directions that this technology can be used to and applied to understand many immunological processes that are right now dependent on extracellular markers and can be used to define many different and very important activities of immune cells, and we're taking it to many different directions. Mapping, using this to map ARG1 positive cells within tumor, and this allows us to reveal that the TREM2 positive suppressive myeloid cells and characterize them, and then later on show that genetic ablation of TREM2 decreases MREG cells and promote immune dysfunction and tumor immune escape. And last but not least, I'd like to really thank the amazing scientists who did all this work and the collaborators. So I'll start with the collaborators. Ton Schumacher, Amos Tanai, Sergio, who didn't show his part of the work, and Miriam Erad, and amazing scientists from the lab, PhD and postdoc, who actually made all this happen, are different grant supporters, and thank you for listening.
Thank you, Ido. That was so exciting. Thanks for sharing this fabulous work. It was very, very enjoyable. I think before we leave, Elina will remind us how people can ask you questions. I saw already there are some questions, Ido, so you will be answering.
We toast again.
Thanks again. Thank you for this great event.
Thank you, Ido. That was a fantastic talk, and thank you also for the toast and reminding us what we have accomplished as well on the 2020. Yeah. Yeah, so we'll, as Carla said, we'll direct everyone to ask questions via Twitter. And remember, questions can be also asked in an asynchronous manner. So basically, if you are not joining live and you will be watching this talk in YouTube, you can also ask questions. And you can search for the account Global IMMUNOTALKS, find the tweet that says ask questions for Dr. Ido Amit here, and reply to that tweet with a question, with your questions and mentioning hashtag Global Immuno, and Ido will use his own Twitter account to answer the questions. So you can connect with him there. Well, thank you so much again, Ido. And thank you everyone for joining, and we hope to see you next week for SASA's Global IMMUNOTALK.
Yeah, thank you, Ido, and thanks everybody for joining. See you next week.
Cheers. See you next week.
Cheers.